Hey everyone, and welcome to RUBIN'S Mind Lab, where we break down the latest research into what it really means. Ever wonder why sometimes when you are talking to someone, it feels like you are just on the same wavelength? Like you finish each other's sentences or just intuitively understand what the other person is thinking? Well, new research is starting to explore how our brains might actually sync up when we interact. This is the core idea we're diving into today, Interbrain Synchrony. It is basically when the brain activity of two or more people lines up or becomes synchronized. Think of it like two musicians playing in perfect harmony. But instead of musical instruments, it is their brains. Scientists have been studying this for a while, especially when people interact face to face. Now, how do we actually measure this brain-to-brain connection? One cool tool that is becoming super popular, especially for studying interactions in more natural settings, is called fNIRS. It is a way to peek into brain activity without having to be stuck inside a giant MRI machine. It uses light to measure changes in blood flow in the brain, which is a sign of brain activity. And because fNIRS equipment can be somewhat portable, it is perfect for hyperscanning, which is when you record brain activity from multiple people at the same time, often while they're actually interacting. The big challenge in these studies is figuring out how to measure that synchronization. The most popular method researchers are using right now is called Wavelet Transform Coherence, or WTC for short. It sounds fancy, but the idea is to look at how two brain signals are aligned, not just in time, but also in different frequency bands. Now, here's where things get a bit technical. And this paper is really about clarifying those tricky parts. The authors are pointing out that while WTC is super useful, it is not as simple as just pressing a button. There are some assumptions you have to make and some choices you have to consider, especially when dealing with the kinds of signals you get from fNIRS. Think of it like cooking. You can follow a recipe, but understanding why you do certain steps makes you a better cook, right? This paper is for those researchers who want to be master chefs of interbrain synchrony analysis. So, the goal of this paper is to act like a guide or a primer explaining the fundamental ideas behind analyzing these brain signals, especially when you're looking for synchronization between people. They start by breaking down what time frequency analysis actually means. Think of a brain signal like a piece of music. You can look at it as a timeline of notes being played, or you can look at the different instruments and their frequencies. Time frequency analysis lets you see both, when things are happening and what frequencies or rhythms are involved. This is a really important concept they explain. You can't have perfect precision in both time and frequency simultaneously. Imagine trying to pinpoint exactly when a single musical note starts and ends, and also know its exact pitch. If you focus too much on the timing, you might not be sure of the exact pitch, and vice versa. This is the time-frequency trade-off. Then they get into the specific tool, the wavelet transform. Unlike older methods that might look at fixed time chunks, wavelets are like little oscillating waves of varying lengths that can be stretched or squeezed. This flexibility allows them to zoom in on different frequencies and different time windows, which is why they're so good for tracking how brain signals change over time and across different rhythms. The output of this wavelet analysis is often visualized as a scalegram, which is like a colorful map showing activity across time and frequency. It helps researchers see where and when different brain rhythms are present in the signals. To compare two people's brain signals, they use something called the cross-wavelet transform. This is where you take the wavelet analysis of each person's brain and compare them. It helps identify when both signals have activity at the same frequency and time. But just having activity at the same time and frequency isn't enough. You need to know if this connection is stable and significant. That is where Wavelet Transform Coherence WTC comes in. It normalizes the cross-wavelet transform to give a score from 0 to 1, showing how strongly and consistently the two signals are synchronized. A high WTC means they're really in sync. And importantly, WTC can also tell us about the phase of the signals, which is like the timing within the oscillation. This can reveal if one person's brain is leading or lagging the others, which is super interesting for understanding dynamic interactions. Now, they connect all this to fNIRS. They explain that fNIRS signals are a bit slow because they measure blood flow, not direct neural firing like EEG. This means there is a natural limit to how quickly we can see brain activity changes, and many of the interesting frequencies are also where we find physiological noise, like breathing. This leads them to talk about cleaning up the data, because fNIRS signals can have a lot of noise. Researchers have to do a lot of pre-processing, like filtering. But they warn that filters, while necessary, can also mess with the timing of the signals, and even introduce their own fake rhythms, which could accidentally look like brain synchrony. Deciding which frequencies to focus on is also critical. They mentioned that the most relevant frequencies for neurosynchrony and fNIRS are quite slow, usually below 0.09 Hz. This is because of the slow blood flow response, and because faster frequencies are often dominated by noise. All of this then influences how experiments need to be designed, because WTC analysis involves looking at signals over a certain time window. You can't pinpoint synchrony at a super precise moment. If you are looking at slow frequencies, you need longer blocks of data. They even mention something called the cone of influence, which is like a buffer zone at the beginning and end of each experimental condition, where you can't trust the synchrony data. This means if you want to study synchrony during specific tasks, those tasks need to be long enough. And of course, they touch upon how to tell if the synchrony they find is real or just a fluke. They talk about comparing the observed synchrony to what you'd expect by chance, or by swapping participants' data around to see if the synchrony disappears. They also discuss that while the more LED wavelet is common, there are other types of wavelets that might be better suited for brain signals, though these can make interpretation trickier. Finally, they emphasize the importance of being clear about how the analysis was done. Since software can have default settings that affect the results, researchers need to be transparent about the specific parameters they used so others can replicate their findings. While WTC is popular, they also briefly mention other ways to measure synchrony, like using different types of transforms or looking purely at phase relationships, suggesting that WTC is just one piece of a larger puzzle. So, what is the big takeaway here? This paper is a deep dive into the nitty-gritty of analyzing brain synchrony with fNIRS. It's essentially telling researchers, be careful and be precise. The core message is that understanding the tools you are using, like the Wavelet transform and Coherence, is crucial. You have to be aware of the limitations, like the time, frequency trade-off, and the noisy nature of fNIRS signal. They're really pushing for researchers to actively work on separating genuine brain-to-brain synchrony from shared noise or artifacts. This means careful pre-processing, smart experimental design, and rigorous statistical testing. The authors aren't just pointing out problems, they're offering solutions and guidance. They want to help build a more standardized, transparent, and reproducible way to study how our brains connect. This is super important because if we can't agree on how to measure interbrain synchrony, it is hard to compare results across studies or build solid theories about social interaction. What I love about this paper is that it demystifies a complex analytical technique and frames it within the broader goal of understanding human connection. It's not just about the math, it's about the science of how we relate to each other. So, what does this mean for us? It means that as research in this area grows, we can expect more reliable findings about how our brains work together. It might lead to better interventions for social disorders, or perhaps even tools to help us communicate and collaborate more effectively. The next steps for researchers will be to continue refining these analytical methods, perhaps developing even better ways to distinguish neural synchrony from noise, and applying these robust methods to understand a wider range of social interactions. What are your thoughts on interbrain synchrony? Have you ever felt that strong connection that seems almost telepathic with someone? Let us know in the comments below. If you found this deep dive helpful, please give this video a thumbs up, subscribe to RUBIN'S Mind Lab for more science breakdowns, and hit that notification bell so you don't miss our next exploration into the human mind. Thanks for watching, and we'll see you in the next one.